Welcome to this lecture on the blood clotting system, a crucial topic in human physiology. Have you ever wondered how a simple paper cut stops bleeding on its own? Today, we are going to explore the fascinating, life-saving biological machinery that makes this possible. We will build our understanding from the ground up, looking at both the biology and the mathematics behind how our bodies patch up injuries. To start, let us build an intuitive understanding of how blood clotting works. Imagine a garden hose with water flowing through it. If a hole suddenly appears, water sprays everywhere and you lose water pressure rapidly. To fix it, you might first plug the hole with your thumb and then wrap it tightly with strong duct tape to make a permanent seal. The human body does exactly this when a blood vessel is cut or damaged. This brilliant automatic repair mechanism ensures that a tiny paper cut does not cause you to lose all your blood. Now that we have our analogy, let us look at the actual biological components. In our body, the thumb is a temporary plug made of tiny cells called platelets. These platelets rush to the site of the injury to provide an immediate but fragile seal. The duct tape that secures this plug is a strong, stringy protein web called a blood clot. Together, the platelets and the protein web form a durable barrier. This system is the body's overarching process to stop bleeding, scientifically known as hemostasis. Building on the idea of the protein web, we must understand how this web is actually formed. The blood clotting system, scientifically known as the coagulation cascade, is a complex sequence of chemical reactions. It transforms liquid blood into a solid gel, exactly at the site of an injury. This cascade involves various proteins, called clotting factors, which are mostly produced by the liver and circulate in the blood in an inactive form. When a blood vessel is damaged, these factors activate each other in a domino-like chain reaction, rapidly amplifying the body's response to the injury. Following this domino-like chain reaction, we reach the ultimate goal of the cascade. Ultimately, this cascade converts a soluble protein, called fibrinogen, into insoluble fibrin threads. These fibrin threads are the actual duct tape we discussed earlier. They weave through the initial platelet plug, trapping red blood cells and creating a durable, solid clot. This clot effectively seals the wound, preventing further blood loss and providing a safe scaffold for the tissue to fully heal over time. Without this final step, the temporary platelet plug would easily wash away. Having seen how the clot forms, it is crucial to understand why this system is so vital to our survival. Without the blood clotting system, even the most minor internal or external injuries would be fatal due to continuous blood loss. This system is absolutely vital for maintaining blood volume and blood pressure. If blood volume drops, blood pressure drops, and the body can no longer pump blood effectively. Therefore, the coagulation cascade is necessary to keep oxygen flowing continuously to critical organs like the brain and the heart. Understanding this vital system helps us make sense of real-world medical conditions. In the real world, we see the devastating effects of a faulty clotting system in diseases like hemophilia. In hemophilia, a missing clotting factor means the domino effect breaks, and patients can bleed uncontrollably into their joints or organs from minor bumps. Conversely, understanding this system allows doctors to prescribe life-saving medications known as blood thinners, like warfarin or heparin. These medications intentionally slow down the cascade to prevent dangerous, unwanted clots from causing heart attacks or strokes in high-risk patients. Now that we understand the biology and clinical importance, let us look at the mathematical representation of clotting kinetics. To truly understand how fast a blood clot forms, we model the activation of thrombin using a simplified enzymatic rate equation. In this formula, V represents the velocity or rate of thrombin production. The symbol K is the rate constant, showing how efficiently the enzyme works. The term XA in brackets represents the concentration of activated factor 10, the primary enzyme. Finally, prothrombin in brackets is the raw material waiting to be activated. If the concentration of factor XA increases due to a massive injury, the rate of thrombin production will increase dramatically, leading to much faster clotting. Let us apply this formula to a concrete example. Suppose a patient suffers a minor cut. Blood tests show the concentration of activated factor XA is 0.05 millimoles per liter, and prothrombin is 1.2 millimoles per liter. The rate constant, K, is 10. We want to find the initial rate of thrombin production. We substitute 10 for K, 0.05 for factor XA, and 1.2 for prothrombin. Multiplying 10 by 0.05 gives 0.5. Finally, multiplying 0.5 by 1.2 gives our final velocity of 0.6. This means every second, 0.6 millimoles of thrombin are generated per liter of blood, ensuring enough thrombin is available to seal the cut. Having calculated how fast thrombin is produced, we can now determine how long it takes to form the actual clot. Once thrombin is successfully produced, its primary job is to convert soluble fibrinogen into solid fibrin threads. We can find the total time required for the clot to finish forming using a simple division formula. Here, T represents the time required. C represents the total required concentration of fibrin needed to completely stop the bleeding. R represents the continuous rate of fibrin production driven by thrombin. Physically, if the rate of production R decreases, perhaps because the patient is on blood thinners, the time T required to form the clot will increase, which could lead to dangerous bleeding. Let us see this time formula in action with another example. Suppose a stable, life-saving clot requires a total fibrin concentration of 3.0 millimoles per liter to completely stop the bleeding from a deep wound. Laboratory analysis shows that thrombin is converting fibrinogen to fibrin at a steady rate of 0.15 millimoles per liter per second. We need to find how many seconds it will take for the bleeding to stop. We substitute 3.0 for C and 0.15 for R. Dividing 3.0 by 0.15 gives us exactly 20. This means it will take exactly 20 seconds to weave enough fibrin threads to completely seal the wound. Now that we have calculated the kinetics, let us visualize the entire process we have discussed. On screen is a schematic diagram of the blood coagulation cascade. Notice at the top we have two starting points: the intrinsic pathway, triggered by internal trauma, and the extrinsic pathway, triggered by external damage. Both of these pathways converge in the middle at the common pathway, specifically at factor 10A. As we move down the diagram, you can see factor 10A driving the conversion of prothrombin into thrombin. Finally, at the very bottom, thrombin converts fibrinogen into the fibrin mesh. This visual perfectly summarizes how a small initial trigger cascades down into the physical web that saves our lives. To wrap up our lecture, let us review the most important ideas we have covered. First, The blood clotting system acts as the body's internal emergency repair kit, preventing fatal blood loss. Second, it relies on a cascade of clotting factors that rapidly amplify a small signal from an injury into a massive production of thrombin. Third, this thrombin creates a tough, insoluble fibrin mesh that reinforces the initial temporary platelet plug. Understanding this delicate balance of enzymes and substrates is absolutely crucial for medical professionals to treat dangerous bleeding or clotting disorders. You now have a solid foundation in both the biology and the mathematics of the blood clotting system.